Okay, so what I want to do is go over this particular paper. I shouldn't say this paper. I want to go over this particular pathway, and I'm actually going to go through multiple papers to help describe or create a model for what the hell is happening when it comes to COVID-19 patients. So I personally think that it is extremely important to focus on minerals or nutrients that are within the body and how they're getting dysregulated, especially during uh, viral infections or pathogenic infections, because these pathogens are frequently searching for nutrients. The reason why they infect you as a host is because you are rich in nutrients, and therefore they want to use those nutrients to produce more of their own, of themselves, right? So iron is probably one of the most important nutrients, and so the iron dysregulation is probably being employed by a significant number of viruses and other pathogens in order to divert some of those resources towards themselves. Okay, so what I want to do is go over this particular method, or not method, but this particular cellular signaling pathway. And the reason why I want to is because this transferrin, we can see that it is elevated in people with severe COVID-19. Same thing with what's it called, ferritin, that is extremely elevated in people with COVID-19. And also the galectin-3 binding protein is elevated in people with COVID-19. So this is why I wanted to focus some of our attention to this, and hopefully we can learn a little bit about this, especially since this blood clotting can also be attributed to, or could be the result of dysregulated iron or labile iron in the blood plasma. So without further ado, look at this cell, and this cell has to figure out a way to maintain labile iron homeostasis within itself. Okay, so some of the things that these cells do is they will have different types of receptors that are able to catalyze chemical reactions, right? And catalyze the reduction of iron 3 plus into iron 2 plus, which then can be pumped into the cell through different types of transporting proteins, right? So this would be one way to get labile iron into the cell from the extracellular matrix. Now, what could also happen is most of the iron that is flowing through your body will not be in this free form, because this is dangerous. This is going to cause unwanted chemical reactions in the body. So most of the iron in your body that is being transported is being sequestered by these transferrin molecules. So here they look like these little light purple teardrops. And these transferrin protein molecules will, bind to two ferric ions. So these purple ions are the iron 3 plus of the ferric ions, and two of them will bind to each transferrin molecule. And these transferrin molecules are floating throughout your body, helping you maintain iron homeostasis and getting the nutrients to the cells where they belong. So this transferrin will bind with this transferrin receptor 1, which will, just know that it binds to this transferrin receptor 1. And this binding helps convince this transferrin with the iron to be endocytosed into these endosomes. So these endosomes are basically when the cell membrane has formed a bubble from the surface to bring this into the cell. However, it is brought into the cell, into this endosome, and so what happens is the ferric ions then leave this and then get catalytically converted not catalytically converted, but it gets converted into the ferrous ion, which are the red ones, and then these ferrous ions are then able to undergo transportation. And as you can see, these proteins, these membrane proteins, also can do the same thing on the surface. However, this is more dangerous to have all this stuff floating around the surface. But here, they also exist in the endosome, because the endosome came from the cell surface. And here, you can have the same thing happening, where it converts this ferric ion to the ferrous ion, and then it gets pumped into the cytoplasm, where it can then exert its nutritive effects as far as producing hemoglobin or producing the iron-sulfur clusters, which are used in enzymes. So this goes to the mitochondria and produces all these other wonderful things, right? So, but some of the negative, and some of the regulatory mechanisms that take place is these IRP12s. Let me drink 1 second. Okay, so these irons will then interact. Well, one thing they'll do is they will interact with ferritin to be sequestered so that they're not producing these reactive oxygen species. So ferritin would be one of the ways that it gets sequestered so that it's no longer damaging the cell, right? And yeah, don't worry about this too much. Just know that these these ferrous ions are then able to interact with these IRP12 proteins. And what are these? These are the iron responsive element binding proteins. And just know that these proteins normally, without a lot of iron in the system, these responsive elements will bind to mRNA. And this binding of the mRNA can either prevent translation or it could prevent degradation. So they can upregulate and downregulate the translation of these mRNAs for different proteins. So, for instance, I'll do a brief overview here, and then we can go into more detail. But here, this iron-responsive protein is interacting with the beginning of the mRNA is called the 5' end. So it's interacting with these 5' ends normally, and so this is actually preventing these RNAs from being translated into their final proteins. So here we have the FNP and the, how did I phrase that? Ferroportin, right? So I'm trying to see if there is a, so here we have the FNP mRNA, which encodes for this ferroportin, right? So if you have a lot of these free IRP12s, they are going to prevent this from forming, right? So, but if you have iron, the iron is going to bind to the IRP12, which then releases this IRP12 from the mRNA, or prevents it from binding to the mRNA, which then results in more of these FPNs being formed. And these FPNs are what pump iron out into the extracellular matrix if you say have too much within your cell. What was I going to get at? So when you have a lot of iron, it will create or cause the expression of these proteins that can pump iron out of the cell. So that's one thing that it can do. And this is what hepcidin actually does. So hepcidin, this is considered the, what would the word be, the insulin for iron. So insulin helps convince cells to take up or hold on to their glucose. Hepcidin does the same thing with iron, where if you have a lot of hepcidin, it actually prevents these transporters from working properly, so that helps the cells store more iron. So just know that that is what hepcidin ends up doing. Okay, that's a little bit of a tangent. So there's that. There's also these FTH and these FTL mRNA. And so what is FTH? Well, this is ferritin heavy chain and the ferritin light chain. So these are the two, these are pieces of ferritin, and this is ferritin right here. So ferritin is the protein that helps sequester free iron ions. So this prevents the iron ions from damaging the insides of the cell because it ends up taking up the iron and holding onto it so that it can be properly stored. So if you have a lot of free iron in the cell, what happens is the IRP12s will no longer be able to bind and prevent the translation of the ferritin. And so the more iron you have, you actually start producing more ferritin as a way to sequester that iron in the cell. Okay, so that's one other thing that happens. And then we have our TFR1, and what is TFR? So this is the transferritin receptor. So remember, this transferritin receptor, this is what allows transferritin to bring bring iron into the cell through endocytosis, which is what I said earlier. So here, the IRPs are actually on the 3' end, and this is where they actually protect. It actually protects the mRNA from degradation. And I'll go into more detail later. But basically what's happening is the IRP, if you have low iron in the cell, this IRP12 will actually protect this mRNA, which then allows it to be translated and produce more of these transferritin receptors, right? So, but if you have a lot of iron in the cell, it ends up interacting with the IRP12, which then releases or prevents it from binding to the TFR1, which then results in the degradation of TFR1, which means more iron in the cell means you have less of these receptors. Okay, so less of these receptors, there will be a downregulation of the receptors, if that makes sense. Now, what I want you to be aware of is what happens when you have a downregulation of the receptors. You end up having more of this transferritin floating around in the plasma. So keep in mind, this is something that we see with people who have, let me get the cell paper up. So transferrin, TF, this cell paper, which was this ultra-high throughput clinical proteomics reveals classifiers of COVID-19 infection, this transferrin was seen in higher levels in the plasma. So what's something that can cause more transferrin in the plasma? Well, that could be the downregulation of the transferrin receptors. So that is something that could cause that. And keep in mind, what else could be causing that is we know that, what else could be interacting with the transferrin receptors is the galectin-3. So we know that galectin-3 also has the ability to bind to these receptors, but that is another topic to be discussed later. So with that, so you have the transferrin that can be downregulated by having more iron within the cell. You can also have this DMT1 mRNA, and this is not dimethyltryptamine, the drug, or this isn't the, what's it called, not psychotic, what would the word be? Psychedelic, sorry about that. So this isn't the psychedelic, this is a completely different protein. And so this actually also will get downregulated when you have more labile iron ions within the cell. And what was this? So this is the ferric ion reductase, DMT1, and so this is one of the transporters here. So here you see this transporter will help bring iron into the cytoplasm. The DMT1 is also one of these transporters, so this also helps bring iron into the cell. But if you have too much of it, then you start downregulating it. Okay. Now keep in mind, these are not the only proteins that are up or downregulated with these iron-responsive elements. So what I should also mention is that the iron-responsive element is this little loop that you see right here. So these little loops you see on the mRNA, this is the iron-responsive element that this IRP12 is binding to. So this is a protein that has to scan, it basically scans this mRNA, right? And only the mRNA that have these little loop-de-loops inside of them will be able to bind to these iron-responsive element proteins 1 and 2. And so this basically the presence of that specific code that causes these loops are going to be the elements which bind to this protein explicitly. And the position of where these elements are at will determine whether or not they are being downregulated, which is what happens here, or upregulated, which is what happens here. So keep all of that in mind, and sorry that I had to go through so much for this, but what I want to do first is talk about the iron response element. So there's actually, here we have one particular code of the iron response element, but truly there can be many different types of codes. And so I'm trying to figure out what the sequence conservation. So okay, so let me just read through this, and then we can make a little bit more sense. So these aren't the typical, depending on the color. Yeah, don't worry about it. We'll keep on going. So in molecular biology, the iron response elements or iron responsive elements, or the IREs, these are short conserved stem loop. This is the stem, this is the loop. The stem loop, which is bound by iron response proteins, and the IRE is found in the untranslated regions. So the untranslated regions, when you have your mRNA, you will have a section of the mRNA that will encode for the final protein, but you have untranslated regions that are before and after. So the before part is the 5' untranslated region, and then the after part is the 3' untranslated region. Now don't worry too much about that. We'll go over this multiple times if the need arises. So let us see. So the two leading theories describe, actually, I won't go into too much detail with this. Just know that this is an example of the iron response element, and just know that these different letters have different, oh, what would the word be? Every letter, so U and A, those are different types of letters that are normally found within mRNA. So if you have these letters here, A, U, C, and G, those are all going to be just single normal, what would the word be? Those are going to be the normal nucleotides. When you see different letters, that is going to mean that there is a combination or different types of nucleotides that could still fit this sequence. But here you can see this, if a letter is red, that means it is highly conserved. So here you have C, A, G, U, G. This is a very highly conserved sequence that is on the mRNA, which helps tell the iron responsive element binding protein to bind to this loop. Okay, so just keep that in mind. This loop is highly conserved. However, the stem, since it's in the greener region, there's a lot more variation as far as what types of, what responsive elements can, or sorry, what letters they can have to still maintain this. So the stem has less, or the stem has less, or sorry, I should say more variability, whereas the loop has very little variability. So this is, just know that that is what's happening. So those are the iron response elements. So what I want to do is talk about the, let's see, okay, I want to go down to the genes that are known to contain these iron responsive elements, because this can tell us if we see some of the proteins that are differentially expressed in COVID-19 patients, if their proteins within their cells are looking like or are following a profile change that would indicate that these iron responsive elements are to blame, they might look very similar to these. Now, granted, unfortunately, I haven't seen a lot of proteomics data in reference to the endothelial cells themselves. The only stuff that I have seen has been of the blood plasma. So only the proteins that are secreted, I have seen proteomics data on. So proteomics being the study of the, basically the proteins that are in whatever sample you're dealing with, and then trying to quantify the concentrations of each of the proteins. So regardless, I've done another video on that earlier. But if we were to be able to look into the cells themselves, the cytoplasm or the cell lysates, then we could also do proteomics of what is within the cell as well to help us determine if there is intracellular changes in the protein. So unfortunately, a lot of these proteins are within the cell, within the cytoplasm, so I was not able to actually, I wasn't able to, what would the word be? they probably aren't going to be found in the plasma as readily unless a lot of cells are dying, but there's probably a lot other things that are playing a much bigger role. Regardless, what I wanted to bring our attention to was this EPAS1. And what is this? This is the, wait, was that the one I wanted to? Yes. So this endothelial PAS domain-containing protein, what is so special about this? Endothelial PAS domain-containing protein, EPAS1, also known as the Hypoxia-Inducible factor 2 alpha, and this is how I remember it, HIF2A, or HIF2 alpha. This is a protein that is encoded by the EPAS1 gene in humans, and it is a type of hypoxia-inducible factors. So what are these? These are proteins that are somehow able to recognize that you have low oxygen. And when you have low oxygen, these proteins then undergo some form of chemical change. And then since this chemical change allows them to then interact with the DNA where they can influence transcription. So hypoxia-inducible factors are a group of transcription factors, or factors that influence the rate of transcription from DNA into mRNA. Okay, so just know that this is one of them. So what I am going to do is move on to this paper called Iron Regulatory Proteins Limit Hypoxia-Inducible factor 2 alpha expression in iron deficiency. Okay, so I just want to go through this paper to help us understand how are these iron-responsive element binding proteins influencing hypoxia-inducible factor 2 alpha. So, hypoxia stimulates erythropoiesis, which is the production of new erythrocytes or red blood cells. Anyways, the major iron utilization pathway. Oh, erythropoiesis is the major iron utilization pathway. So that's where most of your iron is going to. So we report the discovery of a conserved functional iron-responsive element, IRE. So remember, these functional iron-responsive elements, these are the stems and loops that are on the mRNA that can bind to the iron-responsive element binding proteins. Okay, so and this is in the 5' untranslated region of the messenger RNA encoding endothelial PAS domain protein EPAS1, which is also called hypoxia-inducible factor 2 alpha. Okay, so this responsive element is in the 5' untranslated region. So let's go back to this image right here. If we have our IRP12, which form of these is the 5' version? It is this first set. So the first set, where you have this stem and loop, is at the 5' end of these proteins, which means this IRP12 is actually preventing the production of the protein. So if you have iron, then you are actually going to sequester or remove these IRP12s from the mRNA, and then you're going to start to translate more and more of that protein. So with hypoxia-inducible factor 2 alpha, that is going to be upregulated in the presence of iron and is going to be downregulated in the lack of iron. So through this iron responsive element, the iron regulatory protein binding controls this mRNA translation in response to cellular iron availability. So our results uncover a regulatory link that permits feedback control between iron availability and the expression of a key transcription factor, which is our hypoxia-inducible factor, which is promoting iron utilization. And they also show that the iron responsive elements, or the iron response element that is structurally distinct from, for example, the Ferritin mRNA IRE, iron responsive element, and that has been missed by in silico approaches. So in silico means in a computer. So in the past, let me get back this iron responsive element. So currently we often use computer programs to help us determine or help us scan through the DNA and genetic material and then clue us in on what that genetic material might be doing based on its code. So those are in silico methods. Now, as you can imagine, unfortunately, with the iron response element of this hypoxia-inducible factor, there is variations in it. And so sometimes that can make it difficult to find these responsive elements because there is significant variation in between them. So when this happens, they're saying here that they were not able to find this until 2007 because it was a unique form of iron responsive element. So just keep that in mind that this happened. So what is this if let's say there is a lot of iron dysregulation and you do have a lot of iron that is within the cells, or too much iron within the cells. What can this tell us? Well, this will tell us that you are going to have a lot of this hypoxia-inducible factor 2 alpha be expressed. Now, what is the physiological consequence of having way too much of this hypoxia-inducible factor 2 alpha present within the cell? So what I first want to do is go over hypoxia-inducible factors in general. So they are a transcription factor that responds to the decrease in available oxygen in the cellular environment. So we will skip this, we will skip this. I think we'll just go straight down to the function. So the hypoxia-inducible factor alpha expression in the hematopoietic stem cells, so these are the stem cells in the bone marrow that produces all of the blood cells in your body. This explains the quiescent nature of stem cells for being metabolically maintaining at a low rate so as to preserve the potency or the ability to differentiate into different molecules, the potency of stem cells for long periods of time in the cycle of an organism. So just know that the stem cells have stable production of it at a steady rate, and this is something that can help prevent them from differentiating into different molecules. So that's not too important. So for us at least, the hypoxia-inducible factor signaling cascade mediates the effects of hypoxia, which is the state of low oxygen concentration on the cell. So hypoxia often keeps cells from differentiating. So if you have hypoxia, it prevents the cells from differentiating. However, hypoxia promotes the formation of blood vessels, and this is also known as angiogenesis, right? So if you do not have hypoxia, or sorry, if you are in, I should also say hypoxia means low blood oxygen, right? So if you have low oxygen in a location, that actually convinces the cells that they need blood vessels there. They need to get more oxygen to that location. So it sends out signals to say, hey, we need blood vessels, and that convinces your body to move blood vessels into that area. Now, granted, cancer, for instance, will often have a lot of those signals being expressed, which is something that can redirect a lot of your blood flow to a cancer, because they're always putting out the signals, we need more blood vessels, we are anoxic, or we don't have enough oxygen, give us more blood vessels. So that is something that could be happening as well when it's dysregulated. Anyways, and it is important for the formation of the vascular system in embryos and tumors. So the hypoxia in wounds, so if you get cut, this also promotes the migration of keratinocytes and the restoration of the epithelium. So these keratinocytes, these are the, 90% of the cells in the epidermis, but they are basically, if you think of keratin, that protein, these are the cells that are able to produce keratin, which can sometimes be, can cause scarring, right? So some of your scar tissue, that's going to be a lot of keratin building up. So in general, the HIFs are vital to development in mammals, and deletion of this gene results in death. So they don't even survive till birth. So, let me double check to see what. Yeah, so it's the death of a fetus. So or a spontaneous abortion. So H1F or HIF1 has been shown to be vital in chondrocytes, which the chondrocytes are the cells of your cartilage. The chondrocyte survival, which is allowing the cells to adapt to low oxygen conditions within the growth plates of bones. So this HIF, if you have a lot of it, and well, how to phrase that? So the cells of the cartilage have very low or very little oxygen, so this is helping promote their survival in that location. And it's allowing the cells to adapt to the low oxygen conditions within the growth plates of bones. HIF plays a central role in the regulation of human metabolism. So here is the mechanism. You know what? Yeah, so I'll just talk through the mechanism and then I'll go over this image. So the alpha subunits of hypoxia-inducible factor are hydroxylated, which is when you add hydroxy groups or alcohol groups to it, and they are added at conserved prolines, and prolines are an amino acid. So some of the conserved prolines, which are one of the amino acids of the protein structure of hypoxia-inducible factor, these can receive hydroxy groups onto them. And this is allowing for their recognition and ubiquitination, which ubiquitin is a protein that is used as a tag, which can help the cell determine what to do with another protein. But this ubiquitination, in reference to the hypoxia-inducible factors, will send it off to the proteasome, where it ends up getting degraded by these catalytic enzymes in the proteasome. So this labels them for rapid degradation by the proteasome. So this occurs only in normoxic, or when you have normal levels of oxygen conditions. But in hypoxic condition, this HIF prolyl hydroxylase, which is the enzyme that adds the hydroxy groups to the HIF, this cannot work because it uses oxygen to do this, to basically do the hydroxylation. So if you you know what, I'm going to skip that. So I'm going to go to this image right here. So here, in normal oxygen conditions, this is what happens. You have your hypoxia-inducible factor alpha that ends up getting oxygenated or hydroxylated with proline. Proline hydroxylase will come in, take the oxygen and add it with water to form hydroxy groups, and these hydroxy groups will then go onto the prolines of your hypoxia-inducible factor. Then, after these hydroxy groups exist, this then signals VHL to then add ubiquitin, which are these small proteins. It adds ubiquitin to your hypoxia-inducible factor alpha, which then convinces this HIF alpha to then go to this proteasome, where it then gets degraded into peptides. And so this is how, as your HIF alpha is being synthesized, it very quickly will then be degraded by the proteasome, and so you don't really have a buildup of HIF alpha in your cell. However, in hypoxia conditions, when you do not have enough oxygen, this pathway cannot happen. So what ends up happening is this HIF alpha will then be transported, or will get transported into the nucleus. And this transportation into the nucleus then has it interact with this HIF beta, and then it forms this structure, which then interacts with these HREs, which are the hypoxia, I believe they are the hypoxia response elements. I don't know exactly the name of it, but let's call it the hypoxia response element. Oh yeah, here, hypoxia response element. So this ends up resulting in the transcription of these target genes that are right below it. So this hypoxia-inducible factor, when there is no oxygen, ends up going into the nucleus, interacts with HIF beta, and then this complex will then interact with the hypoxia response elements of DNA, where there's probably multiple hypoxia response elements with DNA, and each one of these will have a gene that they want to be upregulated in response hypoxia. And so this will be a way for the DNA to be transcribed in response to create more erythropoiesis, so the production of new or more red blood cells. It can cause metabolic changes, and then it can cause angiogenesis, or the production of new or more red, or more, what would the word be? Blood vessels. Angiogenesis, the production of new or more blood vessels. So that is what's supposed to be happening when you have a healthy individual, right? So let's continue, because I think this will be important. So HIF1, when it is stabilized by the hypoxic conditions, it upregulates several genes to promote survival in low oxygen conditions. So if you have a lot of this, you are surviving in low oxygen. So just keep that in mind. Keep that in mind. So these include glycolysis enzymes, so the breakdown of glucose, which allow ATP synthesis in an oxygen-independent manner. So this is basically convincing the cells to go to undergo different forms of consuming, or how to phrase that? It's basically creating a different metabolic pathway which allows you to survive with less oxygen. So the cells are then using different ways of generating ATP, which is the energy currency of the cell. The cells find another way to do this. So glycolysis is the metabolic pathway that converts glucose into pyruvate and a hydrogen, and the free energy released in this process is used for the high energy production, or the high energy molecules ATP. So the glucose being turned into pyruvic acid, this in of itself is going to produce ATP, and so by doing this, you are allowing yourself to produce the energy that you need without any oxygen. So, and also, the vascular endothelial growth factor will also be upregulated in response to hypoxia, and this promotes, and this is a growth factor or a protein that promotes angiogenesis, or the production of the blood vessels. So H1F1 acts by binding to hypoxia-responsive elements in promoters that contain the sequence NCGTG, where N can be either A or G, so don't worry too much about that. Just know that these are the responsive elements that are found within the DNA, and then this can help direct these hypoxia-inducible factors to the DNA to bind, which then promotes the production of new mRNA. So, let me see if there's anything else that we should talk about here. So, I think maybe we'll have to go through this. So it has been shown that the muscle alpha or A kinase anchoring proteins, or the MEN ACAPs, organize E3 ubiquitin ligases, affecting the stability and positioning of hypoxia-inducible factor inside its action site in the nucleus. So depletion of this M ACAP, or the disruption of its targeting to the paranuclear, also known, or the in of the cardiomyocytes, so the muscle tissue of the heart. These regions alter the stability of H1F and transcriptional activation of genes associated with hypoxia. Thus, compartmentalization of the oxygen-sensitive signaling components may influence the hypoxic response. Okay, so I don't know if I should go into all of this. Just know that I've done another video on nuclear factor kappa beta, or kappa B. I've shown that nuclear factor kappa B is a direct modulator of HIF1 alpha expression in the presence of normal oxygen pressure. So I think if you want to read this for yourself, go right ahead. This is the hypoxia-inducible factor Wikipedia page. So what I also want to do is talk about the repair or regeneration. So in normal circumstances after injury, the hypoxia-inducible factor 1 alpha is degraded by the prolyl hydroxylases. So after injury, you will have more of these H1Fs that are being produced, but then the prolyl hydroxylases eventually do get to them and destroy them. However, scientists found that continued upregulation of H1F1 alpha if you have too much of this H1F1 alpha through PhD prolyl hydroxylase inhibitors. So if you inhibit this prolyl hydroxylase, this regenerates lost or damaged tissue in mammals that have a repair response. So, and the continued downregulation of H1F1 alpha results in healing and scarring responses in mammals with previous regenerative response to lost tissue. So the act of regulating H1F1 alpha can either turn off or turn on the key processes of mammalian regeneration. So what sounds like here, and what sounds like here is if you have dysregulation, you might end up not being able to heal properly. So just know that is what might be happening. So yeah, so with that, just know that this hypoxia responsive element is very important in the production of or in healing. So with that, let me see if there is something else that I want to go over. I think that is it. So what I want to talk about right now is it sounds like a lot of the people who are catching COVID-19 end up getting into this hypoxic state. However, they are completely unaware of how hypoxic they are. Now, what could be causing that? Well, to me, what makes sense is that if you have this hypoxia-inducible factor that is greatly upregulated in all the cells of the body, what that's going to end up doing is making it so that you don't recognize the lack of oxygen nearly as much, right? So your body is actually able to survive without as much oxygen, or you will feel fine without the oxygen. However, what is being described with people who end up dying from hypoxia, it sounds like they're living their best life, they're doing just fine, maybe they have a slight cough, and then they just collapse because they've lived with too little oxygen for too long, and they just collapse and die within a few minutes, right? So what makes sense to me is that when the people are getting this pneumonia and are losing the ability to absorb oxygen well, their body has been producing so much of this hypoxia-inducible factor from the excess of labile iron that it is protecting their bodies from recognizing that you have very low oxygen. So I should look into a little bit how these hypoxia-inducible factors end up resulting in feeling fine in low oxygen environments. But they probably are playing a very large role because people who have mutations in hypoxia-inducible factors, this could result in them not feeling well in different low oxygen environments. And I don't think that I don't have another thing up for this. So this is what I'm imagining, is that COVID-19 comes in and it finds some way to throw an absolute wrench into the iron regulation of the system. It's doing this because it wants the iron to leave the liver and go into the environment so that it can use the iron for its own generation, right? Iron is a nutrient that will be necessary for the stabilization and the catalytic activity of a lot of these viral proteins. So they want to release a lot of iron and get it into the cells of the body, right? So it will want you to release it from the liver and get it, or release it from the hemoglobin somehow, and then that release will allow it to get into other cells within the body, okay? So when this iron gets extremely upregulated, and so you have a lot of this labile iron within the cell, you'll also have a lot outside of the cell, but you'll also have a lot within the cell as well. This is then going to convince your body or the cells to produce a lot of this hypoxia response, or sorry, the hypoxia-induced, shoot, H-I-R, hypoxia-inducible factor 2 alpha. Sorry about that. So you will produce a lot of this throughout your entire body. And when you're producing a lot of this throughout your entire body, you end up being put into a state where you will feel fine when you have low oxygen. So you will feel as if things are great with low oxygen in your body because you just have so much of this hypoxia-inducible factor that is being produced. And this is, when this happens, it's almost as if you can't, let's say you can't feel pain, right? And if you aren't able to feel pain, then you aren't able to avoid a stimuli, or you won't be able to avoid danger as easily if you do not have pain receptors. So if you don't have pain receptors, you end up damaging yourself over and over again, which I believe is the cause of a lot of the damage due to leprosy. Regardless, pain is important, and being able to sense and recognize this, recognizing damage, this is a very important part of survival. If we have too much of this hypoxia-inducible factor 2 alpha in our system, we might theoretically become desensitized to low oxygen environments. And if we're too desensitized, then if the oxygen gets below what is necessary to even survive in the first place, then we're screwed, right? We get low enough where we just die, rather than it dropping and then us becoming aware of our low oxygen levels. Then we can, freak out, go to the hospital, get everything that we need done, get an oxygen breathing tube in to try to restore our oxygen to a healthy amount. But if this is happening right here, where we are put into this very low oxygen environment with a lot of this hypoxia-inducible factor, we're screwed. And I guess what I have to mention is, I'll just leave it at that. That might be a good mechanism by which all of this is happening. And this could also play a role in we'll leave it at that. I don't think I have too much more to add. So with that, I will leave the papers, or what would the word be? The citations to the papers down below in the description if you want to read this for yourself. But this is my thesis, that iron is playing a pivotal role in convincing our bodies that we are fine. Everything's fine. Don't worry about it. But yeah, things are not fine, right? Anyways, with that, thanks for watching, and hopefully I will see you tomorrow.